Hi again, this is Lance Capel, and this is Bio 515. Today I'm doing my presentation on molecular basis of skeletal muscle contraction. Right here I have the different types of muscle tissue. We have skeletal muscle, smooth muscle, and cardiac muscle. If you see here in the diagram to the right, we have the sarcolemma. Then at the bottom, the nucleus. Inside of that, we have the mitochondria. And then we have the myofibrils. Then we have the T-tubules, the terminal systema, triad, and then the sarcoplasmic reticulum. Now, muscle contraction is a complex physiological process that involves the generation of force by muscle fibers. There are three main types of muscles in the human body. And again, that's skeletal, smooth, and cardiac muscles. Skeletal muscles are responsible for voluntary movements such as walking and lifting objects. One muscle fiber or all muscle cells, they usually extend the entire length of the muscle, which means they're long and skinny. These muscle cells can be divided into a myofibril. Myofibrils are then differentiated into sections called sarcomeres, and these are the smallest functional units of muscle cells. This means that they are the smallest section of the muscle cell that can contract and have all the functions of a muscle cell. Muscle cells also have an abundance of mitochondria. Mitochondria make a lot of ATP, and that's needed in the process of muscle contraction. And to our next slide, contraction in the muscular system and neuromuscular junction activation. Muscle contraction is initiated by signals from the nervous system, specifically motor neurons that release neurotransmitters at the neuromuscular junction. The process involves the following steps. Nerve impulses, action potentials, travel down motor neurons to reach the neuromuscular junction. Acetylcholine, ACH, is released from vesicles in the motor neuron into the synaptic cleft, the small gap between the motor neuron and the muscle fiber. Sarcolemma excitation is the next step, and then lastly, ACH binds to receptors on the sarcolemma, which is the cell membrane of muscle fibers, leading to an action potential that travels along the sarcolemma and into the transverse tubules, T-tubules. Calcium release. The action potential in the T-tubules triggers the release of calcium ions from the sarcoplasmic reticulum, a specialized structure within the muscle fiber. Next, we're going to talk about tropomyosin and troponin regulation, cross-bridge formation, power stroke and contraction, and lastly, recovery. In tropomyosin and troponin regulation, calcium binds to troponin, causing a conformational change that moves tropomyosin away from the myosin binding sites on actin filament. Cross-bridge formation. This is where myosin heads on the thick filaments bind to exposed binding sites on actin, forming cross bridges. Power stroke and contraction. This is when ATP is hydrolyzed, providing energy for the myosin heads to pivot and pull the actin filaments towards the center of the sarcomere, resulting in muscle contraction. Recovery. This is when ATP binds to myosin, causing the myosin heads to detach from actin. The calcium ions are actively transported back into the sarcoplasmic reticulum, allowing tropomyosin to block the myosin binding sites again. Muscle contraction is a fundamental process in the muscular system involving the generation of force by muscle fibers. Skeletal muscles responsible for voluntary movements undergo contraction through a series of intricate steps. The process begins with the activation of the neuromuscular junction, where nerve impulses trigger the release of acetylcholine. This neurotransmitter binds to receptors on the muscle fiber cell's membrane, the sarcolemma, leading to an action potential. The actin potential then travels along the sarcolemma and into the transverse tubules, ultimately causing the release of calcium ions from the sarcoplasmic reticulum. Next, the molecular basis of skeletal muscle contraction. We have here actin and myosin filaments, sliding filament theory, ATP, and energy consumption. Actin and myosin are the two main proteins involved in muscle contraction. Actin filaments are thin filaments, and myosin filaments are thick filaments. They are organized in repeating units called sarcomeres, which are the functional units of a muscle fiber. The sliding filament theory explains how muscle contraction occurs at the molecular level. During contraction, myosin heads on the thick filaments interact with the actin filaments, causing them to slide past each other. ATP is essential for the contraction relaxation cycle. ATP hydrolysis provides the energy required for the myosin heads to pivot and perform the power stroke. Next is calcium regulation. Calcium ions play a crucial role in regulating the interaction between actin and myosin. When calcium levels rise, it enables the binding of myosin to actin, initiating the contraction process. Understanding the molecular basis of skeletal muscle contraction is essential for grasping the mechanisms that govern muscle function and movement in the human body. And I just wanted to explain a little bit more. The molecular basis of skeletal muscle contraction revolves around the interaction of key proteins, actin and myosin, which we discussed earlier. These proteins are organized into repeating units called sarcomeres, the functional units of muscle fibers. The sliding filament theory, again, explains the mechanism of muscle contraction at the molecular level. During contraction, myosin heads on thick filaments bind to exposed binding sites on the actin, forming cross bridges. This interaction leads to the sliding of actin filaments past myosin filaments, resulting in muscle shortening. Central to this process is the regulation of tropomyosin and troponin, calcium ions released during the excitation-contraction coupling. They bind to troponin, introducing a conformational change that allows myosin to interact with actin. The energy required for the contraction relaxation cycle is derived from the hydrolysis of ATP. ATP binds to myosin, facilitating the detachment of myosin heads from actin and enabling the recovery phase. And this last page here, it's a little bit of a summary of the muscle contractions for skeletal muscles. So again, muscle contraction is a complex process in the molecular system, primarily observed in skeletal muscles responsible for voluntary movements, initiated by signals from the nervous system. The neuromuscular junction activation triggers a cascade of events, including the release of calcium ions. At the molecular level, the sliding filament theory explains muscle contraction through the interaction of actin and myosin proteins in sarcomeres. This process involves the formation of cross bridges powered by ATP hydrolysis, leading to the sliding of actin filaments past myosin filaments. Key regulatory elements include tropomyosin and troponin, which modulate the accessibility of myosin binding sites on the actin. Understanding these molecular mechanisms is essential for comprehending the intricacies of skeletal muscle contraction and voluntary movement in the human body. Okay. And I have a this little picture here. This is at the top here. The actin potential arrives at the neuromuscular junction, and then ACH is released, binds to receptors, and opens sodium ion channels, leading to an action potential and sarcolemma. And next, you see down here the excitation, and then action potential travels along the T tubules. Right here is the calcium, troponin, and then next to thick and thin filament interaction leads to muscle contraction. And lastly, here at the bottom, as we discussed earlier, the muscle then shortens and produces tension. And these are my references for my presentation, and I hope you enjoyed it. Thank you.